Hi, everyone. I hope you're having a great day, and I'm excited to have you here with us to celebrate our 750th episode of People Behind the Science. I'm looking forward to speaking with our guest, Dr. Alan Saltiel, about his life and research. But first, I'd like to thank this episode's sponsor, Innovative Research. Innovative Research has provided scientists with lab products for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And in our episode today, Alan is going to talk more about his lab, his work, and also his life. So I hope you enjoy getting to know another one of our fantastic people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to be speaking with our guest scientist, Dr. Alan Saltiel. So, Alan, welcome to the show today. How are you?
I'm fine, thank you. Thank you very much for having me.
Well, thank you so much for joining us, and we're looking forward to learning more about you and the wonderful work that you do. But first, let me give our listeners a little bit of background. So, listeners, Alan is Distinguished Professor of Medicine and Pharmacology, the Maryam Ahmadian Endowed Chair in Metabolic Health, Director of the Institute for Diabetes and Metabolic Health at UC San Diego, as well as Director of the UCSD UCLA Diabetes Research Center. He received his bachelor's degree in zoology from Duke University and his Ph.D. in biochemistry from the University of North Carolina. Afterwards, Alan conducted postdoctoral research at the Wellcome Research Laboratories. He served on the faculty at Rockefeller University before joining Parke Davis Pharmaceutical Research in 1990. And he remained there until 2001 when he accepted a position at the University of Michigan. Alan then transitioned to his current positions in 2015, and he has received numerous awards and honors, including the Rosalyn Yalow Research and Development Award from the American Diabetes Association, the Hirschl Award from Hirschl Trust, the John Jacob Abel, Goodman and Gilman, and Pharmacia-ASPET Awards from the American Society of Pharmacology and Experimental Therapeutics, and he was named a Fellow of the Society in 2022. In addition, Alan is an elected Fellow of the American Association for the Advancement of Science, an elected member of the American Society of Clinical Investigation, and also an elected member of the National Academy of Medicine. And today, Alan, we're excited to get to know you as a scientist, of course, but also as a person. So can you tell us, what do you like to do when you're not doing science?
When I'm not doing science? What is that? No, I'm just kidding. No, I do actually have a life outside of science and a family and other interests and things like that. I guess my main thing is physical activities. I'm kind of a sports guy and interested in sports and exercise a lot and still play a little bit of tennis, occasionally basketball, although I think my basketball days are kind of over now. And I like to read and do things on the social side and just other normal things like that. Nothing special. Cook, a few other things.
Fantastic. What do you like to cook in the kitchen there?
For me, the kitchen is kind of like my lab, since I don't work in the lab anymore. So I do all the cooking in my home, which is just now my wife and myself. But I like to experiment a little bit and taste things and multitask when I'm in the kitchen, mostly kind of on the Mediterranean side of cuisine. But I do like to play around a little bit in the kitchen. Again, it's like playing around in the lab, maybe not quite as precise.
You have a little more freedom for creativity, I think, in the kitchen. I like it. Now, you mentioned reading as well, and we love sharing book recommendations with our listeners on the show. So do you have a favorite book, Alan, or perhaps one that you've read recently that you really enjoyed that you can tell us about?
I usually have a fiction and a nonfiction book going at the same time, and I've kind of done that for a long time. And my reading is quite eclectic, so I don't think there's anything super interesting or special about what I do on the fiction side. I often talk to young scientists about career advice things, and I always get asked about advice for books that would help them in the scientific side. So I do have a couple of books that, besides the usual books in the leadership area, I have a few books that I always like to recommend to people that I've read over the last few years that really had an impact on me. One is Thinking Fast and Slow by Daniel Kahneman, and everybody, I think, knows about this book. And this is one of those books I wish I had read when I was younger, but didn't read until I was a lot older. And it's by Daniel Kahneman, who's the founder of behavioral economics. And it's a long, long book. It takes a long time to read. You can read it in segments. I read it over an entire summer slowly because that's the best way to read it. But it really gave me a lot of advice about how to approach problems. So I think that was really an important book to me. Another book that I recommend to people that really had an impact on me is a book called The Inner Game of Tennis by a guy named Timothy Galway. And I mean, it's about the game of tennis, but it's really about so much more than that. And it's about losing yourself in your work and in your play and being in the moment and getting kind of out of overthinking things. So really, like in the game of tennis, the idea is not to think so much about the mechanics once you have the mechanics down. The thing is to think about the ball. And this book had a big impact on me, and I really liked it. It was a great book, and I think a really important book. And then a third book that is also going to sound a little crazy, but I think it's really important for people who are going to lead teams, which is almost anybody who becomes a scientist at some point, is a book called Between Parent and Child by a psychologist named Chaim Ginat. And I ran into this book when I was a young scientist. My wife was taking a parenting class in New York by a woman named Nancy Samelin. And she was a protege of Ginat, who wrote this book about understanding children when they're very young, like when they're going through the terrible twos. The whole idea behind the book is about empathetic listening and understanding the differences between emotional reality and reality. And she made me read this book. I didn't want to, and she made me read it. And I realized when I was reading it that it was really the key to managing a lot of the interactions you have with students and postdocs and colleagues and almost everybody that you work with, like in a 360-degree way, about trying to understand how they see the world and their emotional reality. And the classic thing in this book is about when you have a 2-year-old kid and you give them a glass of milk in a blue cup and they want the red cup. And instead of just arguing with them about how the milk tastes the same in the blue and red cup, you have to first decide if it's worth having the fight about the cups. And then the other thing you need to do is just kind of empathize with the child and say, I understand you really wish you could have the blue cup. I feel so bad for you. You really love that blue cup. I use this technique all the time with my students and colleagues and postdocs and things, just understanding and validating the feelings that they have about a situation, even if you can't change it or you don't intend to change it. So anyway, that was really a useful book for me. I read it a long time ago. I still go back and look at it every once in a while.
Well, Alan, these are outstanding recommendations. Thinking Fast and Slow, The Inner Game of Tennis, and Between Parent and Child. Listeners, you can find these on our website afterwards. And we've talked about life outside the lab, but Alan, I'd love to talk about your science as well. I know you are doing really cool work, but how do you describe it to people who are outside of your field, or maybe people who are not in science at all?
The inner gist of what we're doing in the lab is trying to understand how energy balance occurs, energy in versus energy out, and basically how cells decide, especially cells that are active cells in metabolism, like cells in the liver or in fat tissue, how they decide whether or not to take up and store energy or burn it or release it for other cells to use in response to hormones or nutritional cues that come up during fasting and feeding, and then how the cells respond to conditions of metabolic stress. That is both what I call anabolic stress, which would be like obesity or caloric overload, or catabolic stress, which would be like starvation or cold. We study the pathways in cells that control the metabolic activities of cells, so how they make these decisions. That's kind of how I describe it. Does that make sense?
Absolutely. I think that's an accessible way for people to kind of relate to the work that you do without getting too far into the details, which we will do later in our conversation. But let's take a moment to talk about motivation. I know as a scientist, it can take a little extra motivation to get through the day sometimes if you're stressed out or you have so much to do. So do you have a favorite quote or a saying that really keeps you motivated, Alan?
I do. And I think a lot of the things I think about when you get to this is to remind people to focus on the work. So my favorite quote is really from the Talmud. And it's basically something like this. This is a paraphrase of the quote, but it's not your duty to finish the work, but neither are you free to neglect it. And I think the idea behind that is that it's about the work itself. And it's not necessarily about the rewards you get from the outside or the recognition that you get or anything like that, or even the results so much. It's just about the work. It's about the doing of the work. In our world, it's about the doing of the experiments, and it's about the questions. And it's about thinking and attacking the questions and going on to the next question. So it never ends. We don't expect to finish, but we expect to keep going.
I love that. And I love the application of science, just thinking about doing your part, contributing to this bigger enterprise. You may not be able to answer all of the questions in your career, but you're moving the field forward.
Yeah, you're adding a brick to the building, basically. You're trying to build knowledge. You can't complete the entire building. If you're lucky and work hard at it, you can add a brick or two.
Definitely. And I think role models can help you on your journey as you're building this building of knowledge, adding those bricks one at a time. So when you think back about your career, Alan, are there particular mentors or role models who really rise to the top of your mind as having a big impact on you?
I've had quite a few role models and friends and advisors from whom I've selected different aspects of their approach to things to really help me to get through times and get through decision-making periods and things like that. But by far, there's been one person who had the biggest impact on me, and that was the mentor that I had when I was a postdoc, Pedro Cuatrecasas. So Pedro was a fantastic scientist and leader, both in industry and academia. He was an immigrant. He was born in Spain. His family was forced to flee in the revolution, spent time as a child in South America, came to the States when he was about 12, was a brilliant student, went to medical school at Washington University, was a resident and scientist at Hopkins, went to the NIH for a while, and made some very, very important discoveries. One of his biggest discoveries was a technique known as affinity chromatography, and it was the basis for all affinity kinds of techniques that we use in science. And he decided to focus his career on utilizing these technologies they developed to answer questions in endocrinology and hormone biology and what we now call the signaling in cells. And Pedro was a spectacular scientist, and he decided to move to industry in the 70s. And he moved to North Carolina to take over the R&D at the Wellcome Labs in Research Triangle Park. And this was kind of a shocking thing in those days for somebody who was a scientist of the first rate to go into industry. But he really wanted to do something different and wanted to develop and discover drugs. Burroughs Wellcome, which was the company he went to in those days, was a little bit different than what we know of today. It was really a science-based company. And Pedro was a really unique leader in the area of industrial research because he had a very academic approach to drug discovery. In his mind, it was all about building knowledge and just increasing our understanding of how disease mechanisms worked. And that, in his mind, was the best way to approach developing drugs. So his big focus was to follow the science. And he had a big impact on me. So I went and worked in industry with him for 10 years, which were extremely important to me. And I learned quite a lot while keeping my science going at the same time. And then I went back to academia, but I really learned a lot from him. And I learned about the importance of life and work balance. And I learned a lot about the importance of the focus on science and the focus on the work and not getting sidetracked by distractions and external recognition and those kinds of things. So he's still around and lives in San Diego, and I see him occasionally. And just a really spectacular scientist and wonderful guy. He's the person that really had the biggest impact on me.
Oh, it sounds like you met a wonderful mentor at an important part in your career. And I know you've had a particularly interesting career path, and I'd love to get into some of the details. So, Alan, can you take us back to the very beginning and tell us what first got you interested in science, and then maybe walk us through some of these key moments that really helped you get to where you are today?
I think I was a slow starter when I started in science. I was always interested in high school in science. And when I was an undergrad, I was a zoology major at Duke, and I was interested in science and especially in the pathways and metabolism, because I love the intricate network of pathways that were involved, all the feedback loops and things that were involved in metabolism and signaling. But I didn't really think that I was smart enough to do research. I mean, I thought you needed a level of creativity and insight that I really didn't think I had. But I wasn't sure what I wanted to do, and I thought I would just go to graduate school and check it out and maybe find something. I didn't have that much confidence in myself at that time. And I think I started to discover slowly when I was a graduate student, and more so when I was a postdoc, that I could kind of put the steps together and figure out what the next experiment was. I started to understand about this desire to build blocks and try to build knowledge. It took me a while to really get it. And I figured out at some point that I could really do it. I mean, I wasn't that bad at it. So I started to get more confidence. And then by the time I guess I was in the middle of being a postdoc in Pedro's lab, that I kind of self-discovered that I thought I really did have it. And I was energized by it and excited by the questions. And I realized that that's really, really what I wanted to do. And I would never stop doing that. So I think I made that commitment then. But it was kind of late, I think, for a scientist, because I was already far along in my career. And I started my first academic job at Rockefeller. And when I finished my postdocs, I was directly into the academic world, writing grants. And Rockefeller was an amazing place of science, probably not the greatest place for assistant professors, but it wasn't a place that really supported assistant professors, but it was a great place to be around great science and have great resources and people. But I really enjoyed it. It went really well. I did well with respect to getting grants and papers out. And we're starting to study the signaling pathways from the insulin receptor when I was a postdoc. And in 1990, Pedro, he had been the head of R&D at Wellcome. He moved to Glaxo in the late 80s. And then he had some run-ins with the management team in the UK and decided to leave and go to Parke Davis, which was an old pharmaceutical company in Michigan, which was in Ann Arbor. And they had been around for a long time. And I wouldn't say they were slipping, but they were not doing contemporary stuff in molecular biology at that time. And he decided to go to Ann Arbor and become the president of R&D at Parke Davis. So I was in New York. He was leaving North Carolina. We went through a few things around that time. He went through a few other opportunities, but ended up at Parke Davis. And when he was on his way to go there, he called me and asked me if I was interested in joining him. And he was going to start this new thing. And first of all, I thought, no way, I'm not leaving New York and I'm not leaving academia. But then he got me to go and visit. And I'd never been to anywhere in the Midwest before. And I didn't know anything about Ann Arbor or Michigan. And when he got me to come and visit, I went and visited and gave a talk. It was in November. It was cold and windy and gray and just awful. Weather was awful, but it was him. And I could see when I was there that he was going to build something really exciting. And I had young kids then, and it just seemed like the right move for us. So we just decided to move to Ann Arbor in 1990. I was still pretty young at that time. And I took over as the head of a new department, which I started from scratch, and I reported directly to him. So I knew that was going to be okay. And I knew nothing about drug discovery, zero. I knew nothing at all about it. So all of a sudden, I was on all these committees for developing and discovering drugs, and I learned about different areas of science and aspects of drug discovery I knew nothing about. And I kept my lab running at the same time.
Oh, wow.
I brought postdocs with me from New York. I hired new postdocs, and I built a department of about 110 people in cell biology, and we worked in diabetes and obesity and cancer. We developed some important drugs during that time. A drug for cancer called the MEK inhibitor was the first drug developed in that area. We developed a diabetes drug at that time, and we did some other things. And it was just an exciting 10 years. And I really loved Ann Arbor. I became an adjunct professor at the University of Michigan and had a lot of colleagues there and friends, and was really integrated into the life of the town. And my kids were in school, and that's, of course, how you meet people through your kids. So we were really integrated. So in 2000, when Warner Lambert Parke Davis was subject to a hostile takeover by Pfizer, we went through a period in the summer of 2000 of integration. And I could see very clearly that this new integrated company was not really going to be a new integrated company. It was just going to be Pfizer. And it was just not for me. It was not going to be the same culture. So I decided to make the leap back to academia. And I really wanted to stay in Ann Arbor because my kids were in school and I liked it there. And luckily for me, the university had just decided to build a new institute on campus with a new building, a new institute called the Life Sciences Institute. And I decided to move. They recruited me to the institute. I became the first employee at the institute and then the director. And then I recruited lots of faculty and staff and stuff like that. So for the next 15 years, I was really engaged in running this institute and integrating it into the university, recruiting great scientists, great staff, creating our own culture at the institute. And I think it was a great time. And I had a really, really fun, great time doing that. It was really spectacular and it was challenging, but I really enjoyed it. And during that time, I kept my lab running too.
And then what ultimately brought you to UCSD, UCLA back in 2015?
Two things. One is the big challenge of establishing the Life Sciences Institute was kind of receding because we were well established by that time. And there was the draw of the weather to Southern California. big part of it for us. And then the idea of a new challenge and more of a focus on just a diabetes center kind of thing at UCSD, less administrative responsibility and more time to spend in the lab. So I think that was the big draw, but I'm happy I did it. It's a different kind of a place here, but it's beautiful and the science is amazing at UCSD. And so we really had a good time.
Oh, certainly. And I know moving your lab or starting up a new lab is no small feat, but you know that you can count on trusted partners to get you all of the supplies that you need to get your research back up and running. And we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of ELISA kits, which offer high sensitivity, specificity, and can be used for a wide range of applications. Innovative Research ELISA kits are manufactured in their lab in Novi, Michigan, and they're shipped directly to your location. They are a valuable tool for researchers and clinicians who need to detect and quantify specific biomolecules. Visit www.innov-research.com to view their entire line of ELISA kits. And going back to our guest, Alan, I know you have many exciting projects going on at the moment, but if you had to pick one to tell us about in more detail, what is something that you're working on in the lab right now that you think is super exciting?
Well, my lab, we're interested in this question, again, this overarching question about how cells decide whether or not to store or use energy. But we're focused in three different areas here. And it's kind of interesting how the work always takes you somewhere that you don't expect. So my lab is kind of divided into three areas. The one area is trying to understand how inflammation controls cell metabolism and obesity. And the second area is how cells decide about their disposition of energy, like when they decide to store or burn energy, especially as glycogen. And that's the one area that we're interested in. Glycogen is like a starch of sugar, and it's a way that we store sugar in cells. And we think that glycogen is a signaling molecule in addition to being just an inert form of energy storage. But I would say the most exciting thing right now is this work that we're doing on trying to understand, well, it started anyway, trying to understand how cells decide to take up nutrients from the blood into the cell, especially sugar. And this is under the control of insulin, which is the most important hormone that goes up in the fed state and controls blood sugar. So we've been trying to understand how insulin instructs the cell to clear the sugar from the circulation and go into the cell, the transport of glucose or sugar into the cell. And as we've been dissecting the pathways that are involved in this thing, we stumbled on a protein that's kind of at the nexus of the signaling of the regulatory pathways for the uptake of sugar. And we've been interested in trying to understand how it's regulated. And we made mice in which we knocked out the gene that encodes this protein. And we expected that these mice would have a problem with their blood sugar. They wouldn't be able to take the sugar up into the cells, especially the fat cells, the cells we were interested in. But then when we started to characterize and study these mice, we found that the effect of knocking out this gene was actually the opposite of what we expected. And when we put the mice on a high-fat diet, they didn't gain weight. They stayed skinny. And this turned out had nothing to do with the uptake of sugar into the cell. It had to do with something else that this protein was controlling. And it was the mitochondria in the protein. The mitochondria are the little powerhouses of the cell that make energy and burn energy. So this is a classic example of what happens in science. You have a question, you design an experiment, you do the experiment, and then you see something unexpected. And it's not that your hypothesis was wrong. It's just that there's something else that you didn't know about. And that was classically what happened here. This has happened to me many times, and it's frustrating at the beginning. You think, oh my God, I can't believe my hypothesis was wrong when you see that. But then you realize as you dig deeper that it's not that you were wrong. It's just that you were incomplete, that there's something else. There are other questions that you didn't know about. And so now this has led us into a whole new area of biology, trying to understand how the mitochondria are controlled in response to this protein. The name of this protein is RAL-A. And in response to this protein, how the regulation of this protein by insulin and by inflammation and by other factors is controlling the mitochondria to decide how much energy we burn or store. That's something I'm really excited about because there's still so many questions to answer here.
Absolutely. It sounds like this discovery sort of unlocked a new area of research. And of course, the story is more interesting than you originally thought. And I think that's not often the case in science. A lot of times there are challenges and frustrations that are just challenges and frustrations. So there are these days where everything you feel like is going wrong. So do you have a time that you can tell us about, Alan, when you really struggled with something or you had a major failure that you had to work through in order to just keep things going in your lab?
So many times I can't even number them all. I'll just give one example. There was a time in the 90s when we were trying to understand how the signaling proteins that control metabolism were restricted in space in cells. Because I think one of the big challenges then and still now is trying to understand how hormones have specificity in their signals in cells. And so we were doing this approach called two-hybrid screening, which was just a way to find proteins that bound to each other. And so we were screening libraries of things to try to find new proteins that bound to proteins that we were interested in. And this was useful. We found a lot of interesting new things, and we published a lot of great papers on this. But every once in a while, we would find something that looked unbelievably cool that turned out to be unbelievably uncool. So there's one example of this. I had a postdoc who was working. We had this idea that there must be an important interacting protein for this other protein that we're working on. And we did this screen and we found something, and it looked like it was something so exciting. And so we spent a year trying to nail down what the significance of this new interaction was and what it meant in terms of cell biology. We had grand ideas about a new pathway and a new approach to the control of metabolism. And really, at the end of the day, it turned out to be the kind of interaction that we call true, and unrelated. I mean, it was a real interaction, but it was just kind of a trivial interaction of the technique, and it was not really important biology of the cell. And unfortunately, sometimes it just takes a long time before you finally understand that. And you keep knocking on it and knocking on it. And there are times when we had this kind of problem where we knocked on it and then finally we broke through. But this was one in which we had just had to admit that it was not as interesting as we thought it was. So I think that kind of thing happens to us, to people in science all the time. You get excited and then you realize it's not quite what you thought it was, or it's not anything at all.
Definitely. I think it's a roller coaster of emotions sometimes.
It definitely is a roller coaster of emotions.
And we don't just want to talk about the lows of that roller coaster. I'd love to talk about some of these highs, these successes as well. So, Alan, do you have a favorite example of one of these successes in the lab, whether it was a really big win or just something that was relatively small but particularly meaningful for you?
I really have to get down in the weeds to kind of describe it. But when you work on signaling that controls metabolism, it's really complicated. We were really interested in the role of inflammation in controlling hormone action, especially insulin action in obesity, because we know that in obesity, people and animals who become obese become resistant to insulin. And we and many other people have thought that a mechanism that could explain the resistance to insulin was inflammation. So looking through all the inflammatory pathways, we discovered a molecule, a protein kinase that was important in translating this inflammation to insulin resistance. So this molecule came up in response to obesity, and it reduced the sensitivity of cells to insulin. So it looked like a really good candidate to be involved in the inflammatory link between obesity and insulin resistance. And we not only found the molecule, we knocked it out in mice, and it showed the effect that we expected in the mice in terms of their sensitivity to insulin. And then we also looked for a drug that would inhibit this protein, and we found something really interesting that turned out to be an old inflammation drug, an old asthma drug that was developed in the 80s. And we gave this drug to mice, and these mice lost weight and became more responsive to insulin. So it was really an exciting time. Then we even did a clinical trial with some colleagues in Michigan with the drug and showed that the blood sugar in obese people with type 2 diabetes could be reduced by the treatment with this drug. So it was really super exciting time when we took the whole discovery from the idea of inflammation all the way to a clinical trial over the course of 10 years. It was really exciting. But one thing happened in the course of that was really interesting. So when we first discovered this pathway and this drug, I was giving a talk at a meeting at Cold Spring Harbor, and I was explaining the role of this inflammatory protein in controlling energy expenditure and controlling energy and sensitivity to insulin and things like that. And in the back of my mind, there were still issues with this pathway because there were some things that didn't quite make sense. And I kind of just scoffed over them a little bit. And then I got a lot of nice questions. But then in the coffee break, a European woman, she was an immunologist, she approached me And she said, it was really a great talk. It was quite interesting. And I was really surprised that your drug did this instead of doing that, because the details are not important, but inflammation is associated with changes in energy expenditure. And you kind of got the opposite of what I would have expected. And when she told me that, she was talking to me and she said that to me. And I kind of knew this in the back of my mind, but I just hadn't admitted it to myself, that there was a big, big issue with my hypothesis. And she laid it out for me in a very nice way, not in front of a million people in the coffee break, because that's when you ask a question when you really want the answer. You're not just trying to showboat and right. Other people. And I just said, oh my God, your question is exactly right. I don't know the answer, but there's something that we don't know and there's something missing. And I went back to the lab and I got everybody together and I said, we don't really understand this. There's something that we don't really understand. And we took a really deeper dive into it. And then we eventually figured out there was this feedback loop. And it was the feedback loop that was really important that we were manipulating. I know it's complicated, but it wasn't exactly what we thought it was. It was something a little different that is actually much more interesting and really led us to a whole new kind of idea in the regulation of metabolism, which was very exciting to me. And I really give a lot of credit to this person who challenged me with this question, who's from a different field, but just said something that was almost kind of obvious, really. Sometimes you get the best questions from people outside your field.
Definitely. I think these conferences are really valuable experiences to bring people with different perspectives, bring people from different fields together to, like you said, have these kind of quiet conversations in a corner to really talk through or work through some of the big challenges or some of the inconsistent findings that you see in the field. And I think these conferences, these travel opportunities in science are definitely a perk of the job. So Alan, do you have a favorite place that your science has taken you, whether it was for a meeting or to visit colleagues or to present somewhere?
I've traveled quite a lot, as you can imagine, most of us throughout Asia and Europe and the Americas. I guess my favorite meeting maybe was a meeting in Naples that we had, I think, in the early 2000s, and it involved a trip to Positano for a luncheon overlooking the sea. And we went to Pompeii for a little tour of Pompeii. And then we had these beautiful meetings in Capri and then meetings in Naples. So I think that was one amazing trip I'll never forget. I've been back since a couple times, but that was the most beautiful, wonderful trip. And the atmosphere of the interactions was just, we were out on boats in the Bay of Naples and swimming in the grottos in Capri. I mean, it was an amazing time.
Definitely. I think being able to combine these exciting scientific meetings and discussions with this ability to explore a new and beautiful place to be around the world is a really great opportunity. So if our listeners are able to make it over to whether it's Capri or Naples or anywhere in that region, are there particular sites or things that they need to do?
The first time I was there, I stayed with a very special couple, Gianfredo Pucca and his wife, Arita. They have a penthouse apartment overlooking the Bay of Naples. And I know everybody doesn't get a chance to do something like that, but It was just so amazing. So they took me on tours of Naples. I mean, to museum. I couldn't tell you where we went and everything that we did, but it's such a wonderful city. There's so much to see. I think you really need somebody who's an insider to help you.
Oh, definitely. And I think that's one of the benefits in science. Not only do you get to go to these places, but you develop an international network of people who you get to see around the world, whether it's at these different meetings or other opportunities. And I think the people in science are another thing that really make it a unique career. But I think, unfortunately, scientists are often portrayed a particular way in the media as being very serious, very critical, and sort of always wearing a lab coat and working. And I know the research is important, but just some of the funniest and most creative, amazing people that I've met are scientists. And there's often these quirky traditions or just funny stories or wonderful things that happen when you're together. So do you have an example of some of these human moments in science that people may not often get to see that go against these stereotypes they may have?
The scientists that I know and interact with a lot are really fun people. And I mean, they're my friends. So you select people that you like. I mean, I have collaborators and friends, and they're fun people. They're active. They are interested in lots of different things. They're zealots when it comes to their science, but they're also interested in many different things. And when we get together at meetings, we hike and play tennis. We go sightseeing, and we do all the kind of things that you'd expect. And we give each other a hard time. There's a lot of teasing that goes on.
A little bit of trash talk on the tennis court.
On the tennis court, and also when we're on the podium.
And then does your lab have any particular traditions or things that you do that people may not expect labs or groups of scientists to do together?
I don't think anything too unusual. We have lab parties and picnics and do those kinds of things. There's one thing that we do that I think is really important and it may be a little bit unusual, but I have a journal club with my lab every week. And I have a lab meeting every week too. So the separate meetings to do a journal article and then just review the data. But on the journal article meeting, all the students and postdocs are asked to present a paper for the journal club. But at the beginning of that time, I make them give a 5-minute discourse on some historical event or person in science. Because I realized a few years ago that they didn't have a good sense of the history of science and of their personalities and characters and discoveries and what went into different discoveries and how much chance there was and the serendipity that's almost always involved in important discoveries. So I made them do this, and they love doing this. Every week, somebody gets up and gives a 5-minute overview of, it's often about a person and their life and the challenges they faced or how they made a discovery or the discovery of it could even be an instrument or the discovery of a pathway or something like that. So that's been like the greatest thing for us. It's a little bit different, I think. And I think it's really important for the students.
Oh, absolutely. And I think a fun way to, like you said, bring in those elements of history, because I think they are important. Like, we can't forget the history if we want to keep moving forward in the field, and just bringing recognition to some of these people and their stories and the roundabout ways that some of these discoveries happen, and just the chance and the serendipity, I think are really important for people to learn about early on in their career. So when they're struggling, they understand that the people that came before them struggled too. Mm-hmm. Well, I love that tradition. And I know in science, you often have barriers, things that hold you back from answering those big questions that you'd like to tackle. So, Alan, if we gave you everything, things like funding, staff, technology, feasibility, staff, time, whatever the case may be that's holding you back, were not a problem, is there one research question that you would want to solve most?
The thing about the funding thing that holds us back, I mean, I'm well funded, but I think for all of us, it's the time we spend raising money and writing grants that takes us away from the lab. Some of the grant writing is useful because it focuses your attention on a specific problem, but we do spend an incredible amount of time trying to write grants to raise money to do our work. The system is probably not as efficient as it could be. The major thing that would happen of having unlimited funding would be having more time to think about the research and focus on the research. I don't think I would change the questions that we ask. I'm very excited and enthusiastic about the questions. We probably would employ more technologies and tools that are available now to asking those questions. We have to be careful about selecting the techniques and tools that we use now because of the expense of some of them. So we'd probably create more atlases using the omics technologies that have become available to help us to get at questions. But I think the questions are still the same. I don't think I'd really change the approach that we take. I think I'd still focus on the same questions. And the questions are like, what's going on in those liver and fat cells when they decide whether or not to store or burn energy or release energy for other cells, and how do cells talk to each other in this process? Those are the questions about which I'm excited.
I like it. I think it's wonderful to hear that you're able to work on these questions that you're most excited about. And I think they do have a tremendous impact on people today, right? Because these questions about obesity, diabetes, these are huge problems in society today.
Yes, indeed.
It's wonderful to hear how you would scale up operations to tackle these questions with unlimited resources. And for listeners out there who might be on their own scientific journeys, we love to share advice with them on this show. So, Alan, when you think back about your career, is there one piece of advice that really sticks out in your mind as a really helpful piece of advice that someone gave you at some point that you can share with listeners today?
I think people need to find their own way, and people in science need to find their own approach. Everybody doesn't approach it and do things the same. Some people are focused in a laser-like way on a specific problem or even a specific protein or gene. Some people are focused in a laser-like way on a specific technology that they'd like to develop and then exploit. And all these approaches are fine. There's not one that's better than another. And I have my approaches, and there have been times when I've gotten a little unfocused, and then I kind of came back to my main focus, but it was because I was excited about a finding. So I don't really regret it, but it probably didn't help my career as I look back from a higher altitude. It probably didn't help my career every time I got a little bit distracted on something. But on the other hand, maybe it did enrich my views of things in science. So it's really hard to know. I think you have to find your own way, but you have to really find the questions that you're excited about. And if you're not excited about them, they're the wrong questions. When you wake up in the morning, when you go to bed at night, these are the things that scientists do. They think about the questions. And if you're not in that position, you're either on the wrong questions or you're in the wrong business. And it's not for everybody. I mean, I have seen lots of people come through my laboratory and other laboratories and trained. And, there are a lot of people that just don't have the zealotry about the questions. That doesn't mean there's anything wrong with them. In fact, you might think the opposite, actually. But if you don't have the zealotry about the questions, you don't belong in research. It's too hard. It's too frustrating. There's so much failure. It's so hard to move forward and to communicate your results and to be persistent and to get through the ups and downs and hardship. You really need that burning desire to get at the questions. If you have that, it's a wonderful life. If you don't have it, it's a horrible job.
Well, Alan, I think that's wonderful advice. I think having this insatiable curiosity about the work that you're doing drives a lot of scientists. And I love that point you make that there's no one right way to do a science career. So listeners shouldn't be afraid to really chart their own path. So can you tell our listeners, Alan, if they're curious and they'd like to learn more, what is the best way for them to do so?
My lab webpage is Saltiel Lab at UCSD.edu. And I think we cover on that webpage a lot of stuff that we're doing in the lab, and you can get a little insight into the people and links to our publications and everything else that you probably would need if you're interested.
Oh, perfect. Well, listeners, definitely check out Alan's lab website. And Alan, thank you again so much for joining us on the show and sharing your insights with us today.
Thank you very much, Maria. It was really a pleasure.
Well, it was wonderful to chat with you, and listeners, great to have you here with us as well. We hope you'll join us on our next episode of People Behind the Science.